the first time, there would be no risk of infecting the wrong people, removing the problem of delivery once and for all. More useful than a nuclear bomb, it would leave land, buildings, and your own troops untouched. Because of these technological advances, the next 10 years could produce a targeted virus or bacteria of this type. Selecting its victims by distinct racial groups, it would be the world's first ethnic weapon. A biological agent that could successfully bring about a genocide.
Good evening, ladies and gentlemen. Yesterday we talked about the ACE2 receptor theory. Basically, it's that the coronavirus can only enter cells that have ACT receptors on them, which are mostly alveolar cells. A study suggested that Asians have 5 times more the ACE2 expressing cells than white people. So first, I can't believe that I didn't catch this yesterday, because I even looked at it and I didn't catch it, and I feel like an idiot. But so I did mention that it was just from the study that everybody's obsessed with, was just based on 8 adult donors. Okay. What I didn't realize, I thought that it was, where is it? I thought that it was an Asian and then a white male and a white female. But in fact, it was 2 white females. So it's an Asian male is 2.5% and then an Asian female is 0.5%. Now, I heard somewhere, and I don't remember where, that men have 3 times the amount of AC2 receptors than females do. And that might be that the male Asians versus the female Asians, that they have 3 times as much. But if that's the case, then you could assume that a white male would actually have 1.5% of their lung cells would be ACE2, and that would mean that the Asians were less than twice as prevalent. And that is just a minor observation because we don't have any testing for that. So this right here, this whole argument about the AC2, if Asians have more ACE2 receptors than other races and than white races specifically, that is like the biggest contested fact right now in my entire theory, and it's been really frustrating, and I've spent a lot of hours trying to figure this out. But this study was not the main piece of evidence that I used yesterday that I based my video off of. So what I really based my video off yesterday was this. And this is from the 1000 Genome Project. And when I talked about this yesterday, I could not understand it at all. And now I understand it maybe 30%. I don't know. It's really complicated. Look at this. Look at this stuff. Just look at this. Look at this. See how crazy this is? Look at how smart that looks. And smart people, science stuff. I don't know. What does all this mean? Well, this is a sequence, a variant, a nucleotide. Wait, what is it? Nucleotide. Single nucleotide variant. Yes. See how smart I am? I know things. But basically, okay, so basically, so I'm not sure if this is right or not, but I believe it's right. Basically, your DNA is made up of sequences of nucleotides. A nucleotide, then T nucleotide, then C nucleotide, then C nucleotide again, etc. And 99.9% of these codes are identical. So these codes are identical. 99% in every human shares 99.9%. However, there's that 1.1% where there's single nucleotide variations. And this is where a sequence of nucleotides is the same, except for there's one letter that's changed. And when that happens and more than 1% of the population has one of those variants, that's when it becomes an official one that they study. So these are all sequences. This is a sequence, this is a sequence, this is a sequence. That they identified that when people have this single nucleotide variant, that they have an increase in ACE2 receptors. So we don't know, like, does this variant increase your ACT receptors by 5 times and this just increases it by 2 times? We don't know. Like, I mean, there's probably a way to know if you understand how to look at that crazy data, but I don't know. And maybe the people that made this know. So we'll get to that in a second. But basically, the way that I see it is that, like 100% of them have this one, 98% of them have this one, 82% of them have this variant, and it kind of goes, and you can see each one. Do 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 do. So the people that made this thing, and I don't know who made this, and I don't know what it's about, so your guess is as good as mine as if it's whatever, but this is from the 1000 Genomes Project, and I don't know who put the data together, but I do know that of all the studies that I have, the 1000 Genome Project is the most authoritative, the most reliable, the most accurate. So if it says that these people have ACE2 receptors more, then I would say that it's probably true. And so, yeah, they went through based on those sequences and identified that these people have the most AC2 receptors, and these people have the least. Okay. So okay. Right. So now we're going to talk about we're going to talk about this in a minute. This is the study of the year, basically. This is the study that some Indians, like Eastern Indians, came out, all these cool dudes, and they said, hey, we think that this is airborne HIV, basically. So they're the ones that came out and said that, I mean, that's not what they said, but they said that there's evidence that that is possible. Okay, so first, before reading their argument, I want to read the counter argument to that. And basically their argument is that there's 4 inserts. Here, maybe I should just explain it. There's 4 inserts that are unique to nCoV that are not presented in other coronaviruses analyzed. So this is what sets this new virus apart, are these 4 inserts. And it just so happens that these 4 inserts match with the human, HIV proteins, especially, and they're identical to residues mapped in the HIV GP120, and now that is vital. So we'll get to that in a minute. But this right here is showing where these inserts are identical to HIV, okay? So, on to the argument against it. Okay, so they're saying that this program that they use to search for this is not what it's designed for. So looking at 300 million viral genomes for a sequence that short means you're bound to find something, and other scientists have pointed out in the hours since the Indian paper was posted with similar overlaps. Just as strong may be found in a wide variety of viruses and also bacteria, protists, fungi, fruit flies, and plants. They go on to say, now we're talking about the GP120 protein, which I said is really important. So the spike protein allows the coronavirus to recognize the AC2 receptor and invade mucous membrane epithelium, while the GP120 allows the HIV virus to recognize the CD4 receptor and invade the CD4 T cells. The GAG protein on HIV host 4th matching sequence recognized by the Indian team is the interior of the virus. So, if the hypothesis were true, you might expect that the 2019 nCoV strain to be able to infect T cells or recognize the CDR receptor. And that basically means that it's identical to HIV. But they say there's no evidence so far that the 2019 nCoV can infect T cells, or that it can infect any cells expressing CD4, or that it can infect any cells which don't express ACE2 and can't be infected by other known coronaviruses. Okay? So for me, that doesn't make me feel very much better, that argument. Hold on a second. Basically, it takes 3 to 6 weeks for T cells to start dropping after you've been infected with HIV. And this is not HIV. Even if it does the same thing that HIV does, it doesn't mean it does it in the same exact way. So if this virus is able to do the same thing that HIV can do, which is infect your T cells. Okay, and to explain that really quick. Following rapid loss in the first few weeks of HIV infection, the number of CD4 cells in the blood falls slowly, often over a period of 10 years or longer. The exact mechanisms by which CD4 cell depletion occurs are not fully understood. So, HIV, not very well understood still. Been studying that for 40 years, okay? So, you can't tell me that they know whether or not this thing can infect T cells after 4 weeks of studying. All right, because we don't even know if they are checking T cell levels to, are they worried about this? Are they looking into this? Or are they just, focused on keeping people alive? Who knows? So GP120, which is what we're talking about, right? GP120 is the important thing, is essential for viral infection as it facilitates HIV entry into the host cell, and this is the best known and most well-researched role in HIV infection. So if someone was indeed trying to modify this virus to be able to infect T cells, and the reason why that's important is T cells are what control your immune system. So with HIV, it kills all your T cells, your immune system stops working, and then a regular old virus can kill you. And that's what AIDS is. So basically, if somebody was trying to take the coronavirus and modify it so it could infect T cells, what they would do is add the parts that allow it to infect GP120, which would probably involve splicing the actual HIV DNA with the other DNA. And we have this.
The Felix program is to determine whether a piece of DNA, a sequence of DNA on a computer is genetically engineered or not. IARPA has funded many different performers across the US to take a crack at this problem when we're all taking very, very different approaches. You can slice these kind of signatures of engineering in a number of different ways. So some organisms are 80% AT, some are 80% GC. What this means is that if you take the DNA from different organisms and glue them together, and you're counting, the A's, C's, T's, and G's, eventually, if you're looking at DNA that's been glued together from disparate sources, you'll see some major swing in those statistics.
To investigate.
So this is what we're looking at. Did somebody put these in here? And even if they did that and it failed and they can't get into the GS120 or GP120, I don't know what it is. If somebody could have attempted to get the virus to be able to infect T cells, and this would just be evidence of that attempt. So it's just potential evidence that this is in fact a bioweapon. Okay, so and this just explains how HIV works. But I think we have a good understanding of that. This is HIV, okay? So I forgot to get a thing of coronavirus, but coronavirus looks different, very different, but it has the same exact shape, and then it has the same exact things that GP120, this is the part on the coronavirus that is allows it to enter the ACE2 receptors. Okay. So these 4 sequences are what make the novel coronavirus unique from the previous strains, and they also happen to be found in the GP120 molecule. And the GP120 molecule is what happens to allow the HIV virus to enter T cells. So that's a pretty big deal. All right. Okay, so if we want to probe into this whole bioweapon thing further, we want to look at, is it possible that this virus did occur naturally? And so, first we would want to say, an ongoing outbreak of Middle East respiratory syndrome coronavirus suggested that this group of viruses remains key threat and that the distribution is wider than previously recognized. Although bats have been suggested for the natural reservoir of both viruses, attempts to isolate the progenitor virus of SARS-CoV from bats has been unsuccessful. Now, I forgot to make this tab open, but there's a super fun conspiracy theory from 2003 where these Russian scientists were convinced that the original SARS virus was a bioweapon from the United States made from measles, and I can't remember what the other virus was. And it was just ridiculous because, like, then we found out that it's a coronavirus, and so those were a completely different strand type of virus. But so it's been looked at before that maybe SARS was bioengineered, and maybe it was. Like, it could have been that the coronavirus was isolated in the 90s, and then they turned it into SARS, and now they've turned it into this. So, yeah. 99.9% sequence identity to RS3367 and uses ACE2 from humans, civets, and Chinese horseshoe bats for cell entry. Preliminary in vitro testing indicates that sorry, I'm tired. I've been staying up. Oh my god, I've been working on this video forever. And I'm tired. Okay. Our results provide the strongest evidence to date that the Chinese horseshoe bats are natural reservoirs of SARS-CoV. Okay, that's the point of that. There's a study that says that, and I don't need to explain it to you, but that's just the thing. All right. Okay, so now this is what happened. I came up with this entire theory, and I did all this work, and then I went on 4chan, and I made a stupid post, and somebody responded, and were like, I'm about to ruin your entire life, because here's the thing. And basically, they sent me this study, and this is what it claims. So, in current severe global emergency situation, it is imperative to identify vulnerable and susceptible groups for effective protection and care. Recent studies found that the 2019 nCoV and SARS nCoV share the same receptor, ACE2. In this study, we analyzed 4 large-scale data sets of normal lung tissue to investigate disparities related to race, age, gender, and smoking status in the ACE2 gene expression. No significant disparities in ACE2 gene expression was found between racial groups, Asians versus Caucasians. I never realized that rhymes. Asians versus Caucasians. Age groups 60 to 60 and below 60, gender groups male versus female. However, we observed significantly higher ACE2 gene expression in smoker samples compared to non-smoking samples. And there is an explanation for why in the first study that was just one person, one Asian versus 2 whites, he could have been a smoker, and smokers and Chinese people are more well known for smoking. And so that's like Chinese smoke way more. I should have researched how much more they smoke than white people, than Americans. but they smoke more, and so that could be why SARS is impacting them more. Let's just Google this. How much more do Asians smoke? Or how about Chinese smoke than Americans? $320 million people. Okay. How many people live in China? I don't remember. 1.3 billion. So only 320 million out of 1.4 billion. That's still not that high. And like, okay. How many people smoke in America? So it is much higher, right? Because that's... I don't want to do ratios, I'm tired. But it is higher. But, you know, that doesn't really explain this. Okay, so this is my argument. This is a preprint. This is not a reliable study at all right now. This is preprint, which means it hasn't been peer reviewed. Which means that just, like, I don't remember, who does it say? It's like one guy that this is like, I mean, they do have graphics and such a little bit. I mean, you can just see how simple this study is. And I read through it and it was kind of hard to explain, but basically I don't trust the study. Mostly because I have evidence to the contrary, which is this, which I'll get to in a minute. But these are just the references. This just seems to be like a reactionary study trying to argue against the study that everybody's talking about. Yeah, it's just this one guy. There's this one guy that says, you're all wrong, this guy's wrong, you guys are all idiots. That's this guy. He's in the Department of Environmental Health Sciences, Arnold School of Public Health, University of South Carolina. All right, so that's this guy, okay? And he posted this on Prepost, which is just a preprints, which is just basically non-peer-reviewed, random things that haven't been like officially looked into. So we don't know how legit this is. But I will tell you, see, now this is why. So we're going to get into this. But what nobody is reporting is that the race of the victims. They appear to be all Asian, specifically the yellow race, formerly known accurately but now politically incorrectly, the Oriental. Thus, 2 coronavirus cases were just confirmed in the UK. Coronavirus UK patient is University of York statement. But if you read the article, both turn out to be Chinese nationals. Similarly, a recent headline of Helsinki Times read, first case of Wuhan coronavirus found in Finland, but it turns out to be a Chinese tourist in Lapland, not a Finn. In fact, as far as I can tell, all of the confirmed cases have been Chinese people. Okay, and then it talks about all these cases that were Chinese people, blah, 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 blah. According to Wikipedia, a total of 774 people died across... This is talking about SARS. And SARS is like 97% similar to coronavirus. So, potentially in line with findings of the Chinese researchers, the mortality rate for SARS in Europe was almost nil. Minuscule numbers of people caught it, and only one of them died in France, where there were an atypically high 7 cases. By contrast, in Hong Kong, the mortality rate for SARS was 17%. In Taiwan, it was 10%. In Canada, it was 17%. But it seems obvious that they were all Chinese Canadians. What with there being so many Chinese people in Canada and documented attempts to help them deal with the virus. In Singapore, 13% of those who caught SARS died out of 238 cases. Other countries had very small numbers of cases, or as with Vietnam, their medical access would have been suboptimal, rendering it difficult to make comparisons. But essentially it can be said that the SARS was an East Asian disease which, like corona, can be traced back to bats. SARS didn't severely impact the rest of the world despite people definitely catching it. It disproportionately killed East Asians. See SARS, the immigration dimension, blah, blah, blah. Many Chinese people believe that the SARS virus could be a biological weapon manufactured by the United States. Okay? So, there's a bunch of stuff that I explained to you. To summarize, which I probably should have written a better summary. And I'll mention this. It's stupid. Just to summarize, if ACE2 receptors are more common in Asians, and you know what, I can't. I should have written down a summary. I can't do this right now. So yeah, basically you understand what I'm talking about at this point. We're just going to take a super sharp left turn and talk about this really quick. Basically, this is a little conspiracy theory going around that China stole the virus from Canada and then accidentally released it. And this is the person that they are saying. They're saying, okay, basically this Chinese guy is a scientist at the Wuhan Institute of Virology, which is the level 4 biohazard lab right outside of Wuhan. And they're basically saying that he was the one messing around with this virus and the reason why it got out. So, first it says that a help wanted post notice was posted on their website on November 18th, 2019, according to which the Institute is seeking to hire 1 or 2 postdoc fellows who will use bats to research the molecular mechanism that allows Ebola and SARS-associated coronaviruses to lie dormant for a long time without causing diseases. So this is what they were researching, supposedly, when they accidentally released it. Or, alternatively, it could be somebody in the government. It could be an American spy. An American spy could have entered the research facility and just released it. I mean, there's all sorts of things that could have happened if it was released from the lab. There's unlimited possibilities. You know, this guy could just be a psychopath, the person there. There's just a million things. And I doubt that if this is what happened, I doubt that the CCP will ever tell us. The Communist Party will probably never tell us. So, but it is possible, and we're going to just go into this. Wuhan Institute of Virology in China find that bats, an antiviral immune pathway called the sting-interferon pathway, is dampened, and bats can maintain just enough defense against illnesses without triggering a heightened immune reaction. We believe that there is a balance between bats and pathogens they carry, says Senior Peng Zhao. This work demonstrated that in order to maintain a balance with viruses, bats may have evolved to dampen certain pathways. So basically they're saying all these viruses exist in bats, but they don't make them sick. So can we just make it so these viruses can exist in humans and not make us sick? But they said, for example, in humans, an activated sting pathway is linked to severe autoimmune disease. Okay, so bats carry viruses, and then these are, this is a Google translated press release from January 18th, 2019, describing the achievements of Dr. Peng Zhao. And then it says bunch of stuff, and this link will also be in the description. So I have all of these links saved for you in a very long list. They're in the order I talked about them in the video, and that will be in the description. So it is understood that Xi Jinping is the pioneer of global bat immune system research. Bats carry viruses but do not get sick. They have not been researched by scientists before. Okay, so that's an interesting statement, right? They have not been researched by scientists before. Bats haven't ever been researched by scientists before? Okay, who knows what that was supposed to mean. But I'm too tired to speculate randomly. In the long run, bats carry the virus without getting sick. It is hoped that humans can learn how to fight the virus, but this is still far from industrialization. Okay, so there's that conspiracy theory. It's an interesting theory. It's a possible theory. And, yeah. So that's basically the video. I have a couple more things to show you. First, I wanted to tell you guys that even if this is not a bioweapon, which it very well could not be, there's a very good chance that it is not a bioweapon. and there's no pandemic and everything's fine, that does not mean that you shouldn't be preparing, okay? You should be preparing. After I did this research on bioweapons, I was like, wow, this is going to go down. Like, people are afraid of nuclear bombs, and it's ridiculous, because this is what you should be afraid of. You should not be afraid of nuclear weapons, because those are probably never going to be used. Biological weapons almost certainly are going to be used. And you need to be prepared for that. Everybody knows that you need to be prepared if a crazy pandemic happens. So even if this is not the case, this should be your wake-up call that, okay, it's time. We need, a year's worth of food. We need all the medical supplies. We need everything. And I'm planning to do a video that basically shows the cost of getting everything you need through eBay and Costco. So you guys can get an idea of how much it would cost to stock up on everything. And so with that, I will show you this really quick video, which was interesting, that I just came across when I was doing the video research.
Member of a white supremacist group, Larry Harris, showed how easily an extremist could obtain deadly germs. Posing as a medical researcher, he contacted a lab.
I ordered 3 vials of bubonic plague, and basically get a catalog off the shelf, reach through, call them up, give them a charge card number, and they send it to me. Plain and simple. Yes, that is Yersinia pestis. That is correct. As far as a person getting into the biological...
So yeah, terrorists can apparently get their hands on viruses and bacteria fairly easily if they really want to. I'm sure it's harder. This was from 2016, okay, that documentary. Actually, I don't know if it was. That was when it was posted on YouTube. Never mind. I don't know when that was, because it does seem like it's a much older documentary. So disregard that. I don't know how old it is, but it's probably harder now. But still, if a terrorist really wants to get their hands on a virus or a bacteria, they can probably do it pretty easily. So be afraid. Be very afraid and get prepared. Okay, I'm telling you that right now. And I have one more video to show you, and it's just because it's an interesting video. So it's like the most interesting thing I came across, and it's about cancer treatment.
She has managed to modify the ricin poison so that it attacks only the cancerous cells in the body, leaving the healthy ones untouched. It does this by recognizing genetic markers. The ricin is attached to an antibody which looks for receptors on the cell surface. Because of a genetic mutation, cancer cells have different receptors from normal ones. The antibody recognizes them and attaches. Only then is the ricin activated to kill the cell.
So there's a thing, people using crazy bacteria to kill cancer. Don't know what happened with that, but it's interesting. So, and it just gives you an idea of this whole how the virus or the bacteria uses its key to access a cell. So yeah, that's basically the video. We got nice and technical. My brain is melted. And I hope that you learned something. I hope that I'm wrong and right about a lot of things. I don't know. I don't care anymore. But please, I don't know how true any of this is. Do not think of me as an authoritative person. I'm trying to learn. I want to know as much as possible. So if you come across anything that contradicts or verifies or anything that I've said, please send it to me. And you can send it to me on Twitter. So you can add me on Twitter, or you can mention it in the comments. So yeah, thanks for watching. I will be adding more videos. I'm adding more. I have another 15 minutes of videos from inside China that I'm going to add soon. I don't know if I have the time to edit it together tonight because it's midnight and I'm tired. So I will talk to you guys later. Bye-bye.